This study delves into the intricate biochemical pathways involved in the formation of complex molecules, particularly focusing on how thermodynamic parameters shift as molecular weight increases. Using Biochemical, we calculated reaction paths to explore the assembly of hexamers from various proteins, analyzing the equilibrium between monomers, dimers, and higher-order oligomers. We assumed multiple biological systems coexist in solution, like LGP2 protein and double-stranded RNA, with their ratios influenced by experimental conditions. By calculating thermodynamic values for each compound, we can assess energy barriers between different states. Let's consider each calculated thermodynamic indicator in more detail. Let's start with the stability parameter Lg_dW, which is presented on the graphs in a logarithmic scale. In this case, a smaller value of Lg_dW corresponds to a more stable biological compound and closer to thermodynamic equilibrium. Accordingly, the higher the value of Lg_dW, the less stable the biological formation is, and the further the biological formation is from thermodynamic equilibrium. In this state, it will be in search of further chemical reactions in order to get closer to thermodynamic equilibrium. Change in entropy in this paper is given as the difference between the mutant type system and the wild type system. Negative values of the entropy change signal an increase in the ordering of the system. The change, dWd, in dissociation energy is also counted as the difference between the mutant type system and the wild type system. Let us begin the analysis of monomeric LGP2 proteins with different mutations, each of which is in solution in a monomeric state. We calculate a set of thermodynamic parameters for each such monomeric protein for the wild type WT-LGP2 and mutant variants MUT-LGP2. The first three monomeric LGP2 complexes in the graph are characterized by either a small decrease in the stability parameter, figure 4a, or a significant decrease in stability, as in the case of the E573A mutation. The remaining three monomeric complexes are characterized by a sharp drop in the Lg_dW values. This tendency of increasing stability at the very beginning of the formation of a biological complex at the stage of a monomeric protein may signal the achievement of thermodynamic equilibrium of the biological system of the monomer even before interaction with the two-stranded region of RNA. But we will continue to study the addition of subsequent molecular subunits, see Fig. 4b and 4c. Note that the increase in stability goes in the direction of achieving an equilibrium state. When the system reaches thermodynamic equilibrium and increasingly actively enters into subsequent reactions, and in this case, we need to add another protein mutant subunit, which also demonstrated in the monomeric state increased stability compared to the wild-type protein. Suppose that another identical mutant protein is added to the LGP2 RNA system. Consider figure 4c for the LGP2 8DS RNA LGP2 biological entity. The normalized pooled values of the stability magnitude are shown in figure 4f in more detail. In the case of the I597S mutation, we see small fluctuations in stability for all three molecular entities. The remaining three monomeric complexes show a sharp decline in the values of log_dW. This trend of increased stability at the very beginning of the biological complex formation at the monomeric protein stage may indicate that the thermodynamic equilibrium of the monomeric biological system is reached even before interaction with RNA. In figure D, we observe a drop in dissociation energy values into the negative range for the intermediate LGP2 RNA formation, marked with red spheres. We will summarize the conclusions and results in a final tables. You can see that the characteristics of mutant monomers differ significantly for those formations that reach thermodynamic equilibrium before the formation of the trimeric complex, with a large number of transitions and barriers. To summarize, it was found that mutations in LGP2 can significantly increase the stability of intermediate complexes, such as mutant LGP2 monomers and the LGP2 RNA complex. Thus, we can determine the stoichiochemistry of biological entities by calculating thermodynamic parameters. The remaining three monomeric complexes show a sharp decline in the values of log_dW. This trend of increased stability at the very beginning of the biological complex formation at the monomeric protein stage may indicate that the thermodynamic equilibrium of the monomeric biological system is reached even before interaction with RNA.